Okay, now let's get back to the original respiratory system again. So the surface area of oxygen exchange in the lungs for diffusion. So because that's what at the end, the diffusion happens. So I brought this picture from Saltzman's book that our lungs at the bottom, it will be composed of alveolus, a small spherical sac, air sac. And then there's a smooth muscle, there's capillaries for gas exchange, and there's elastic fiber. And this is the actual picture. So you can see red blood cell, and in the capillary and simple squamous epithelial cells are there. So the cartoon for this is here is inside capillary red blood cells are there, and the capillary is composed of this capillary endothelium, and there's endothelium is based on this basement membrane. The other side this air space, alveolar air space, and there's a surfactant. And you can see the dimension is only about like 2 micron across. So the distance of hemoglobin, the RBC, versus the airspace is only a few micron apart. So let's go back to number of alveoli is about, we know that the instance is about 300 million alveoli. And each size of alveolar diameter is also about 300 micrometer. Then we can calculate the total surface area of oxygen exchange. And you can do this by mathematics. Then it turns out to be about 80 square meters. And you can think of this typical single tennis court of like one side 8 times 24. Oh, like one side single tennis court is kind of comparable. And what's the total volume of the lung space? You remember in the respiratory physiology, average volume of the lung is about like 4 liters. So think about this, the tennis court, this half of the single tennis court, the huge space, you folded it into a 4 liters of into our lung. So every time we breathe, we are actually having the surface area of half of this tennis court. So that huge surface area makes this oxygen exchange through into our deoxygenated blood, and also we are actually sending out used carbon dioxide. So the absorptive surface area in our lungs is enhanced by these very special architecture by many small spherical alveoli producing huge surface area. So you see this a great invention of how makes this huge surface area makes an efficient transport of gas molecules. And let's get into then gastrointestinal tract. The longest section of gastrointestinal tract is a small intestine, and that there comes most of the absorption of nutrients. So how does that look? Is this luminal surface has actually a little bit of this circular folds, and that circular folds contains of course so-called villi, which is this intrusion of the surface area that is covered with endothelial epithelial cells. Of course, this inside the protrusion, you can see capillary gets in and out, and also at the same time, these lymphatics also are there. It's called lacteal. And these epithelium of the intestine, in fact, the pathologist watches with the light microscope, there's a very hazy surface on top, so that luminal part of or apical surface. And because of that, pathologists named it as a brush border. It looks like some small brush, but it's hard to see. And that it turns out to be called a microvilli, and there's a very tiny intrusions, only clearly visible by electron microscope. So this is a cartoon to showing the small intestinal lumen, and that has this a fold intestinal wall. And that folds contains this villi structure. And the villi structure, the apical surface, there's a very tiny microvilli. So next page, I'm gonna try to work with you how much of a surface area can be enhanced. So we know that small intestine is about 5 to 6 meter long, and the diameter is from 2.5 to 3 centimeters. So based on these circular folds on the inner luminal surface and the villi and microvilli, how much of the total surface area is increased relative to a, just assuming just plain tube? And the answer, I wrote it down, is about 0.39 or 0.4 square meter to huge. It's like hundreds of fold, 235. So now here, as a comparison, I put tennis full court as 10 meter by 24, so total 240. So this is very comparable. So our small intestine is like a whole tennis court, like folded it down and then put it into our tube. So let's work on this as a board exercise. Okay, so let's do some board exercise. That's a surface area of human small intestinal tract. So first of all, so that small tract is a just a linear plane tube. Then the length L is about 5 meter, and the diameter D is about 2.5 centimeters. Then what's the surface area? Let's say A1 is the rim, which is a pi times the diameter and time multiplied by the length. So that will be A1 is pi times D times L. So that's pi times diameter as a meter, 0.025 meter, and length about 5 meter. So that is about, let's see, okay, yeah, 0.125 pi or 0.393 square meter. So that's the first. Then how about A2, which is a folded, the real one. So then first circular fold, which is on the side of this, on the luminous space, there's a circular fold. So that is, if you look at the picture from that PowerPoint, so that becomes, let's say, like these circular folds is becoming, originally about this surface becomes a 1, 2, 3. So it's about 3 times. So roughly about 3 times, okay? And then what about this villi structure? So villi structure is, looks like this, and that's about 10 times. And third is a micro villi structure. Is in fact, it's much more thinner like this, and that we can assume about 20 times of increase. So overall, this is about total 600 times. So then A2 will be about 600 times of A1, and you can calculate this is about 235 square meter. So that's our conclusion, and I have to say that this is a rough ballpark figure, and you know, the numbers here may not be exactly true, and in fact there is a controversy. So I wrote it down in the board exercise, the link of a paper which is only 2014, claiming that, hey, the total area of human adult gut mucosa is not order of tennis court, but half of the badminton court. but maybe I don't know whether how important it is, but it's a good idea to go back to check and always be suspicious of even my calculation. You can bring your own ways of solution. Okay, I'm gonna stop it here.